We're back again with HDYO's Breaking Down Barriers series, and we're having another casual conversation where me and my co-pilot Ashley Clark have a easy conversation about an interesting program that's happening in the field, so it's more accessible for our community to understand these exciting developments in HD therapeutics. Um, I'm Lauren Byrne. I'm a HD researcher and family member, and I'm joined with Ashley.
Hi, everybody. My name is Ashley, and I am an advocate in the community for Huntington's disease. And yeah, it's great to be here again for another video.
And we're very excited to introduce Bill Haney from Skyhawk Therapeutics. Bill, could you please introduce yourself, tell us a bit about your journey kind of into the community?
Sure. First of all, what a privilege to be with both of you and with the Huntington's Disease Youth Organization. I admire what you're doing. It's not easy, and you do it with grace and humor, and I particularly appreciate the latter. So, yeah, I mean, I think that my personal background, I grew up in Rhode Island. I grew up at a monastery in Rhode Island, a Benedictine monastery in Rhode Island. I went to Harvard scrubbing toilets to pay for college, and I started my first company there 45 years ago. I was interested in how science and technology could make human life better. In that case, I invented systems for reducing air pollution from power plants that are on every power plant in the world. And I've been sort of working on how you can use science and technology to improve human health and welfare to a first approximation ever since. And some of that is in companies like Skyhawk. Sometimes it's been in nonprofits. So I've started a set of nonprofits to help women and children in the developing world, and some of it's in filmmaking, where we try to talk about subjects that we think will have big international impact in a way that's approachable and ideally engaging. So I got into drug development. First, I helped people start companies in drug development, but I didn't run any. And then my father died of cancer. And I had a college roommate who was the head of MIT's, the biggest global cancer research center in the and he had a discovery in his lab and he thought could really transform cancer. And he asked me if I wanted to work on it. I said, I really don't. It sounds cool, but it will take forever and cost a fortune. And, I'm getting a little long in the tooth. I'm not sure I want to do that. Maybe we should find somebody younger. And he said, but your father died of cancer. And I said, that is a good point. So I got involved in drug development initially working on cancer drugs. And we have a series of cancer drugs in the world treating patients, I'm happy to say. And then I started really focusing on this extraordinary RNA revolution that's been taking place and how it could affect neurological disease. And my sister-in-law, my wife's sister, Kathleen, and a series of folks discovered a novel science about the way you could use RNA to build pills that would change this course of disease in a way that was extraordinarily patient-friendly and scientifically revolutionary. Historically thought to be impossible, there is a terrible disease called spinal muscular atrophy, which is at the time had no disease-modifying therapeutics, very, very challenging, discoverable at birth, fatal by 20, really, really tough characteristics. And she and others figured out a way to do what had been considered impossible and to invent a pill, prove it worked in animals, take it to the clinic, prove it worked in patients, and actually get it globally approved. And that journey taught us that such a thing could be done. And most patients, even if there is an alternative of gene therapy with a 10-hour operation and the country will pay the $3 million, or lumbar injections, quarterly, even when that's a choice, that's pretty tough on patients. And so the idea of a pill that goes, you take it a day, and if you don't like it the next day, you stop. And felt very appealing to go after lots of diseases. We started Skyhawk to do that, to go after rare neurological diseases, and to build a set of tools that would be able to transform, neurological challenges. And we started it with the folks who'd done this one previous example, and a gentleman who won the Nobel Prize for discovering how all of this works. And Skyhawk in our Huntington's programs are a result.
Wow. What an amazing journey. Thank you so much for sharing so much of kind of your personal journey as well. I very much relate to my dad had Huntington's disease, and it definitely fuels my passion for getting involved in research. Nothing, no bigger motivator than a family connection, I think. A lot of big terms going about. You mentioned we're going to dive straight in because I think some of our community would love to really understand that nitty gritty as well. Let's start with some like, what is mRNA? Like,
Yeah.
you meant,
Well,
yeah.
there's a central dogma in biology. DNA makes RNA. So think of DNA as the permanent copy of our genetic structure, an individual genetic structure, and RNA is a temporary copy. And RNA makes protein, and we're built of protein. So, and mRNA is in that continuum, and it's the way the instructions are carried to the spliceosome to build the protein successfully. So if you thought about the challenge of most disease, and in this case, Huntington's disease, it's ultimately a challenge of protein. You don't have the protein structure that it's not ideally constructed. And one choice has been historically, people have tried to drug the protein itself. And that's essentially the history of all drug development for the last, pick how long you want to go back. The Egyptians were working on breast cancer. What we're saying now is if we go upstream of that, rather than having a misconstructed protein, if we can fix the instructions so we don't misconstruct it at all, that's a lot easier. Think about your kitchen. If you build it improperly and the stove and the sink are in the same place and you decide to rip it all apart and fix it, that's a lot more expensive and challenging than it is to just build it, make sure that the blueprints are right the first time. And the idea of drugging RNA is that you can fix the blueprints the first time. And I think it's a revolutionary idea. It was just historically considered impossible. Phil Sharp won the Nobel Prize for being able to do it, for discovering how you would think about this. And the first work in this was called ASOs, and they were one way of drugging it, but they can be fantastic, but they have great limitations. The next change was gene therapy. As we know, intrusive operations. Then the vaccines, the mRNA vaccines, which was essentially how can we teach the body to build an antibody against COVID, for example. And now we're into the notion of pills, which can be globally available. You don't have to go to the hospital for all the time. If you take them and they're designed properly, they're out of your body in a day or two. So if for some reason they're not working, they're not permanent like gene therapy. They're very inexpensive for the countries to afford, so it's easier for insurance. People in rural areas can have continuous application. So we're just trying to find ways to make it cheaper, easier, simpler, less intrusive for the patients while providing, transformative benefit.
So Skyhawk released some really interesting data last year about the phase 1 trial. I think a lot of people are new, hearing about Skyhawk for the first time. It's like came out of nowhere. So, could you give us a bit of an update on kind of what those results were?
So the history. So the notion is we get mutant huntingtin protein when we have a misconstruction at the mRNA level. You get protein forms that are both the healthy and an unhealthy form, and the unhealthy form ultimately can cause disease. So for a long time, people have been working, I think, quite hard to try to figure out how you could correct that. And the first thing was lower the mutant form of the Huntington's, perhaps both forms of Huntington's, but certainly the mutant form. People started working on gene therapy as well. It's not likely to be available to the full community worldwide in any way, shape, or form because it's really, really expensive. And I think a lot of people, quite understandably, are nervous about a 10-hour brain operation, 10-plus-hour brain operation. Then there have been pills. There's two companies, us and a company called PTC, that have invented pills. Skyhawk has invented what we believe is a better pill, and the data you're referring to shows not 30% reduction in the mutant huntingtins, but 72%, and shows that not only do we reduce mutant huntingtins, we reduce something called PMS1 that kind of amplifies the negative problems of Huntington's disease. And so
what's PMS1?
Yeah, it's PMS1 is another gene, and if you modify it, you can reduce what are called somatic mutations. So you can reduce what's called the CAG repeat.
What somatic mutations?
So You're changing yeah, it's an interesting question. Essentially, there's an amplification in the genetic code transmitted in the RNA that causes and extends Huntington's disease for patients. So they get a misprotein and they get essentially an amplification effect. PMS1 affects the amplification effect. So
I think going back to remind people what somatic instability is, we've had a few sessions and I'm sure we can link to those before. We had Triplet Therapeutics in the past and Locus 23. So when you get a genetic test for Huntington's disease, you're giving a CAG repeat length of say 41 repeats. But what we find, and that's just from testing the blood, but different tissues in the body actually might have a different range of repeats in the cells in that area. And in particular, areas that are really affected in Huntington's, like the part of the brain that is really vulnerable, has much higher repeats. And there's been evidence from genetic studies in real patients where other genes that could have an impact on how people get the disease, we call them genetic modifiers. PMS1 is actually one of them. So the idea is like you can have other genetic genes that influence how early or late someone could get the disease. And PMS1 is part of this family of genes that are really important for managing and keeping our DNA healthy. So there's this kind of link that this DNA repair and maintenance is potentially linked to also this kind of phenomenon that people have somatic instability. So much more repeats in the brain, and that can be worse for certain people. So when you I can't remember if you have more PMS1, would that make somatic instability work? That would make it reduce it. So PMS1 is, would you say, potentially protective?
Yeah, I think first of all, thank goodness I've got you as a translator. So thank you. I can see why that your guide through this thicket for people, and it's fun to listen to you describe it so well. And the answer is yes. It's a sense effect in a way, and if you can reduce it, it turns out, at least the academic literature, is if you can reduce it as little as 10% at the same time you're modifying mutant Huntington's, you get a real benefit in disease delay. And
okay, so more PMS1 would lead to more somatic instability, and we think somatic instability is bad. That's the kind of theory. Are you with this, Ashley?
Yeah, I am following. And I'm not going to lie, it's not easy. It is a lot of terminology and stuff, but I think I am keeping it up. So I am. Yeah, I think I am.
But if you have questions, Ashley, this is why we have,
Oh, no, don't worry, I'll interrupt. So, well, I think I'm getting there. It's so Skyhawk has developed this pill, or that's what, they're working on this pill, to develop this pill, and it not only goes after the mutant Huntingtin, but it also goes after, I'm going to say the wrong thing.
PMS1. Yeah.
Yes, it's also going after that. And if we can get the two of them, then that's a good thing, because the PMS1 causes this somatic instability.
Yeah, I think you've done.
I mean, I'm keeping up. I
no,
am.
you got the gist of it. I mean, that's the by this chance.
Somatic instability is bad.
We believe so. Yeah.
So we believe. Yeah. So the fact that we're going after the two with this one pill, that's obviously a very good sign.
Killing two birds with one pill.
Yes. One pill, two birds.
I think, and we also know, I mean, I think that Lauren said it very wisely. The data is pretty compelling on PMS1, but there's some slight debate. But we're pretty darn sure that if you can reduce HTT by 75% safely, that's way better than 30%. And the data that we've published shows that we were able to do that so far in healthy volunteers. So that's for the field, that's the best data that's ever been published that I'm aware of. And that you can also modify PMS1 is likely to be very good, but even if it's irrelevant, dropping HTT 74% is going to be a real advantage.
Yeah.
So what you're saying, your drugs kind of potentially more potent. So is a kind of has a bigger whack for less amount of drug, which could be good because
You
then
know now, yes. I mean,
yeah.
I don't think I think it's just an objective fact. The alternative is publish their data. We publish our data. I
Mhm,
think it's an objective fact that we have much lower reduction with the data we have today.
mhm.
So the trials are
So
ongoing.
how do you get a pill to or a small molecule to affect Huntington protein and the DNA, like the RNA? Like what
Like, how are
I
you
think
targeting just that with a pill?
Yeah, it's a complicated process, and I think you have to have very, very, very gifted chemists and biologists to do that. And we were fortunate. The folks who invented the first drug ever approved as a small molecule, as a pill to modify mRNA, we were fortunate to hire those people to come figure it out here. So they've already proven you can do it, and they did it. And there are patients around the world taking that pill very successfully right now. And those patients, it's so safe that that's been approved by the FDA, for example, and worldwide, daily dose pediatric for life. So they have infants taking that pill every day for their whole life. And so far,
but
that's
in Huntington's, like how are we like targeting it? I think sometime I think some people have heard the word splicing a
yeah.
lot, which can confuse a lot of people. Do you want to take a stab at that? I'm here to help.
What do you tell us here, Lauren?
Don't look at me for this splicing. I did like
The way that I think about it for myself, and the way the gentleman who won the Nobel Prize describes it to me. And if you see the movie, it's designed to make this all a lot simpler. So there's animations that you can see, and I think you'll find it simpler. Um, but if you imagine that you wanted to make a rope that was 100 feet long, but you only had three 33 feet long pieces, so you had to tie them together, to get 100-foot long piece. Those places you tied it together, or if you spliced it together, would be the splice sites. And not surprisingly, that would be the place if you put a lot of pressure on the rope and you pulled really hard, the places that would be most likely to have challenges would be the places that you spliced it together. So we've discovered that that process creates challenges in the way you might imagine. And if you're going to fix a challenge, the place that you go to fix it can be the splice sites. And so
Cool.
there are a modest number of splice sites. In this case, it's called the 5' splice site. And we're modifying the mRNA at
5' is just the start.
just the start. So we're modifying at one of these splice sites. We modify the RNA so that the protein that you make downstream will be all healthy and not diseased.
so.
Yeah.
Can I ask a question, right? So I'm fit. Lauren, I don't like him. He steals my like way of doing analogies. Like normally took
No,
the pill here.
I've never heard it described like that, and
It's really helpful. So help me out here. So I'm picturing this rope that's tied to get these three sections ropes tied together. And let's say the first knot is splice site 1 and the next knot is splice site 2. So if this rope is like your lifespan, okay, if we go to splice site 2 and that's where we're targeting with the pill, does that mean any Huntington's like symptoms or effects on my body that's there? So let's say I'm thinking about my dad, and I'm thinking about, he's got the involuntary movements, and he's got that jerkiness, he's got those movements, and then we get to splice site 2 where the rope's tied together and he starts taking that pill. Does that then mean that hopefully he won't get, the loss of speech and the loss of the ability to swallow and the other things that comes with Huntington's, and this pill is going to stop making that protein, and then he'll just get like old as normal from that splice site 2. Have I explained that very well,
I
Lauren?
think you might be merging two kind of things there, Ashley. So
Ideas together.
definitely, I think the one question you're saying is like, is this drug going to be helping stop symptoms progressing? Like, is it disease modifying? Will it prevent disease? And then the other thing about the splice side, that's really about when Bill was trying to explain, we have this kind of dogma of biology. You have DNA, is the, our genetic blueprints. That's really important, that stays in the closed in the nucleus of the cell, and we don't want to mess it up. So a copy is made, and that's the RNA, or the mRNA is kind of another step of that process. So we normally think of it as DNA, RNA, and that makes the protein, but actually this messenger RNA is kind of like an edited version, like a finesse version of the first copy from the. Would that be accurate, Bill? Like,
is it like draft 2?
It's like the draft that goes to production.
Right.
It's like
Gotcha.
it's like the proof. It's been proofed.
Okay.
Splicing is like where they cut those unnecessary bits out.
Okay.
But because of
Gotcha.
that, like biology, the drugs that Bill's talking about can target those parts because they're vulnerable.
Yeah.
And that's what he was trying to say about these like parts in the messenger array are actually quite vulnerable, that then you can manipulate it and try and. So when the drug interacts with those splicing part, why does that then reduce all of the protein, or?
We didn't test to, so I guess the short answer is we don't know. What we know is that it reduces the mutant protein by 72% when dosed up to 9 mg daily. And we didn't see any dose limiting toxicity. So we think you could have dosed higher, but 72% is so much better than anybody's ever done before, and very likely to be therapeutic based on what the field knows, that we were ecstatic to get where we were. And and I will say that, and thanks to the Huntington's community, we opened the trial with patients in January. We've dosed now more than 24 patients. They've been on drug. Many of them have been on drug for 3 or 4 months, which is, if there were going to be challenges, when we would expect to have seen challenges. And we haven't seen any challenges so far. We've been approved to now dose for more than a year based on a series of things. And I think the patient advocacy groups in Australia have been fantastic. The clinicians and the hospital sites in Australia have been fantastic.
I was just going to ask where, so this is like a current ongoing trial. Where is it taking place?
Yes, it's current ongoing trial. And right as of today, it's taking place in Australia. This summer, it will open in New Zealand as well. And then this fall, it will start opening around the world in 20 countries. And that includes countries in Europe, in Africa, in Asia. And I think that we found that the patient, and as an example, and I think it's a wonderful contribution by your community, the number of patients coming to hospitals to try this has been very, very large. And because of that, the trial is going very fast. So we want to treat 150 patients on the phase 2/3 and 400 in the global phase 3. And if those 400 come, in 6 months, then we get the data in 6 months. And if we're right, then we can move to have approval for everybody to have access to this. If it takes 4 years, then it takes longer. And the enrollment, the number of people coming has been so fast that we're going faster and faster.
And how long is the trial running for you to like collect that data? And
we don't know exactly. So where we are is the first piece, the first phase has been approved to be 12 months plus 84 days, which is some interpretation of 3 more months. So, and then if the patients are doing well, they usually have a choice to stay on if they like. The second piece starts at 12 months and likewise can extend. And we have to watch the data. And the third piece will be the same. It's a drug that the present data says the half-life is 30 hours. So it's essentially almost entirely out of your body in 2 days. And to date, the data has been very compelling, and it's been running in cyno studies, now for 7 months long, and it's all been healthy there too. So best as we know, we have the best data of any drug ever in the easiest way to get it ever. And it's been safe for everybody, so we just have
So is this a phase 2 trial, or I thought it was a phase 1, but is this now branching into phase 2?
No, you're right. It's branched towards pivotal. So there's 2 pieces, and I think you're right. It's little. I mean, it's a great sign from the Australian clinicians and patient community and regulators. So it started exactly as a phase 1C. So you're right. And it was supposed to be 28 days.
And just to remind everybody about kind of the development, phase 1 is really primarily about safety and trying to get some idea of signal of meeting the target, which they obviously did by lowering huntingtin so well. But when you're getting to phase 2, you're really trying to tease out doses and really show evidence for what we call efficacy. Basically, is the drug actually having an effect on huntingtin symptoms?
Correct. Beautifully said. And so what was to be a phase 1, because it's going very well, has been extended in length to be consistent with what would be a phase 2/3. And because we're doing the study to examine data that you'll be familiar with, although some of your community may be less familiar with, but the VMRI data, the CUHDRS data, the mutant Huntington's data, that data will be gathered. We think essentially this drug has a chance to be approved in Australia in 2026 and in the United States the year and Europe the year after. We don't think that will require things to go smoothly with the drug, and it will require patients to enroll quickly, which they've been doing, but that to continue. And if that happens, we think it can be approved, relatively soon. Never as soon as we'd like, but relatively soon.
Yeah. And have you seen any? Because it sounds amazing. It sounds like you've done so much, and with the history that you have, you have went so quickly through the steps and stuff, and seeing such great progress, having lowered it, 70-odd%. Did you see any side effects or anything to that? Because I do like that you said that, it's a pill that you take, and, if you stop taking that pill within around 2 days, that's out of your body. So this is not permanent. This is not something permanent that, say, for example, I would be taking. But was there any side effects so far in the trial? Because you do seem to be moving quite rapidly through it, which is a good sign. But I do just want to ask.
Yeah, it's a very good question. And I have to say, people who invent new drugs have to be humble about challenges to come. So I can only tell you what we've seen. I don't know what will happen next. So
Totally.
I think you should expect there will be some side effect of some kind, even though what we've seen to date has been modest. It doesn't mean there won't be something at some stage because there might well be. So here's the information we have. We ran a 13-week cyno study. It was safe. We've now started what was to be a 9-month cyno study. We're 7 months in. So far, it's been safe. In patients, we ran a control group that were getting essentially a sugar pill, so placebo, compared to the patients, the healthy volunteers it started with, and the safety was the same. So the people on the sugar pill said they had a headache at the same pace as the people who were on the different doses of the drug. So we didn't see any difference. I can't say there was no difference, but there was essentially no meaningful difference between placebo and the drugs to date. And we've been in patients since January. And so far, what we've seen is very exciting, but there's lots of work to do. And, there's always a possibility of a challenge to come. And on the one hand, I want to share hope, but on the other hand, I have to be humble that we don't know what we don't know. And there could be problems. We just haven't seen them just yet.
So what's the next step then? What can we look forward to in the next year? You mentioned, 2026 potential of approval, but kind of what is the timeline?
Expanding out of Australia and stuff, that's exciting.
I think that in the short run, what you can expect is us to dose the first patients in the phase 2/3 in Australia and New Zealand. That should happen this summer. I think you can expect us to dose the first patients in the global trial. They'll be enrolled for observation this fall. They probably won't get dosed until first quarter next year. So that means not just Australia, New Zealand, but Scotland and Georgia and Canada and various other places. I think we're going to talk to the Australian regulators this summer and in the winter, and we'll see what happens. I think that there seems to be we will have been gathering data in Australia for well over a year by the fall of 26. So we've dosed the first patients in January 25. So we'll have, if we can get accelerated approval, given that there are no disease-modifying treatments available, so it would require that. It would require the data to still be good, and the Australian regulators to consider it to be worth accelerated approval. You could be approved in only Australia at the end of 2026. Then we would continue to be gathering data from Scotland and Georgia and other countries that we're chatting with, and we would share that with the FDA and the EMA, for example, in 2027. What they say is, of course, up to them. And there's some possibility that what they say is the data looks great. you're approved, but you have to keep running the trials simultaneously. And so while you continue, you're approved during that time, and we have to watch the results at the end. And there's a chance they say, we want to be more conservative, and you're going to have to finish those trials. And a lot of what affects that, of course, is the data that comes and how fast people go to the trials. So one of the things that we saw in Australia, the average enrollment per site for previous trials was about half a patient a month per site. And the Skyhawk trials were 1.7 patients per month per site. And so everything's moving fast because more people came to try than had happened before that we're aware of anywhere. And that changes the schedule, because what we want is 125 patients, including placebo, in the phase 2/3, and 400 plus patients in the global phase. And if those 400 patients happen in, obviously it's the faster they're there, the faster we get the data. I'm afraid
which I guess is a credit to the HD community.
Yeah, I think there's no question. The HD community in Australia has been spectacular. The advocacy group's been spectacular. The patients themselves, it takes a certain courage to go participate in one of these trials. They can be inconvenient just because you got to go someplace you weren't naturally going. So a lot of it, there's got to be a partnership between the drug developers, the clinicians, the regulators, and most importantly, the patients.
So you hinted at a film that could help us explain all of the complex things that we went through.
Yeah.
Go ahead, Ashley.
I was gonna say the same. I was like, this is all very new, very exciting. Some people in the HD community may not have heard of Skyhawk, and that's why this video here is so important. But yeah, I want to know more. You kind of dropped that in and just swooped on by. So what was this you said about a movie? Tell us what that is.
It's so I made a movie called Cracking the Code. It is not specifically and exclusively about Huntington's, but it is about the revolution in RNA modulation we're talking about. And it features, on the one hand, a boy born in a one-room dirt floor house in Tobacco Country, Kentucky, so dyslexic he can't fully read now, and his journey to winning the Nobel Prize for discovering how RNA works, and pioneering the insight into how you might imagine drugging RNA so as to fix protein mutations. And on the other hand, it talks about this and he changes that dogma of how DNA makes RNA, makes protein, so that we understand it better. And the second piece is how he starts building drug companies who can really help patients on critical diseases in ways that, I understand if I was a Huntington's patient, I'd be wondering, why the heck hasn't somebody solved this yet? And
we wonder it quite a lot.
I have to say I'm incredibly sympathetic about that. And on a positive note, the film highlights examples where, once that's how diabetes patients felt, once that's how multiple sclerosis patients felt, once that's how spinal muscular atrophy patients felt, there were no treatments. And over time, and I wish it was faster, drug development persevered and found treatments. And I do believe that's going to happen for the Huntington's community. I really do. And so the film highlights a bit of this. And I think is an opportunity to learn about how RNA can work, and about, how drug development can work out. And frankly, that it's really hard. The human biology is very complicated. I want to give you simple, clear answers, but I also mostly want to be accurate. And so have to condition with a bunch of stuff we don't know.
It sounds fascinating.
It sounds like it's going to be amazing, and I feel like it sounds like it's going to be very, even though it's not specifically about Huntington's disease, I think it's going to be great for the Huntington's community, because it's going to help us better understand that science behind it. And I don't know about you, Lauren, but I've seen a massive, I don't know the right word. The Huntington's community is getting so engaged in the research and so engaged with the companies working on the different trials, and they want to learn and they want to know and they want, to get that information. And that's why videos like this are so important, because it shows that, science and community working together can only mean one thing: the hope that we all have that one day something will work. So I think that, the film will help only just further that. So that's very, very exciting. So it is.
Importantly,
Um,
how do we
I
watch
was,
it,
I was
or
gonna,
where?
I was gonna say, like, when am I adding this to my Netflix watch list? Like, how, when's this
Where's it going to
coming?
be? Where, where, yeah.
Well, I think that for you, for your community specifically, we're going to give a free copy for your screening only in June. I think it's
Oh, exciting.
Are we VIPs?
you are definitely for me VIPs. The
I want to get a virtual red carpet.
I'll have to get a dress, and that
No
sounds
problem.
fun, right? Exactly. The uh, More broadly, it will be in theaters this fall in the United States, and then on PBS in America. And then it will be globally available in the late fall, early winter, and we'll start putting it onto the platforms that you're describing, Netflix and Apple and
That's so
Amazon.
exciting. Yeah.
That is amazing. So we're kind of looking at, coming like, international premiere like throughout June and stuff. And we'll link everything. Like, here's me, like we do it. Let's be fair, we all know Jenna's sitting in the background here, and she makes sure everything's linked out and gets out to the community and that. So that's very exciting. So it is. That's
Yeah.
brilliant.
So I think we're coming up to the end, Bill. It's been amazing chatting to you. Really interesting stuff and really exciting progress. We always like to ask, how can the community stay up to date and learn more? Obviously they can go to learn about the kind of the biology of it from the documentary, but in general about Skyhawk and keeping up with the program.
Yeah, well, thank you. I mean, and by the way, please reach out to me or to our team with questions or suggestions. You're so involved, and I do think you're right. It's a partnership between the scientists, the drug developers, and the community. And I think that the government, I mentioned it before we started, I'm here talking to folks in the Senate in the United States, and they listen if you're and regulators listen if advocacy patients are really important to talking to these folks about, hey, look, if it's not the perfect answer, but it's better than no answer, we'd like to talk about it. I think is also very important. And the trials are on clinicaltrials.gov. I'll make sure that it's sent to Jenna and she can put it up on your website. Skyhawk is just skyhawktx.com. Please feel free to follow us. I believe that we're on Instagram and other sort of social media platforms, and we'll try to keep everybody up to date. We are trying hard to go as fast as we can, but it's quality first and speed second, I'm afraid. And so it's most important to get it right. And we'd like to get it fast, but we're focused on getting it right. And we're off
I
to
Well,
like
you're
that you said
certainly
that.
going pretty fast, and
Yeah.
maybe it's because we haven't, I guess, because it seemed like out of nowhere when you came on the scene last year or 2 years ago. So,
Yeah.
but
I
it
feel
feels
like
like you're going fast.
Well, I'm glad
yeah,
to
and I
hear
feel
that.
like he's joined with having accomplished so much that everybody's like just flabbergasted with like how quickly it's moved. But yous have been clearly working on this for many, many years before coming into the like spotlight of the HD community. So it is very exciting, and I do, as somebody who lives at risk of Huntington's, I appreciate you saying that you're going as fast as you can, but safety and making sure it's right is what's important. And you're trying to keep that speed up, but you're also being super careful, because I know that's a concern for some people. Like, what if it comes too quickly and it's not checked out and stuff. So it sounds like you're doing a brilliant job.
But we're doing our best, and you know how challenging it is. And we do. I think part of the story is we just didn't talk a lot about when we didn't have good human data. We thought we didn't want to create false hope, and so that this is an industry. Some people are motivated by different things without judging them. What we're focused on is what is going to be good for patients, and we thought saying something before we really knew wasn't going to help anybody. Um, so, but what a privilege to be with both of you. Thank you for the leadership you're showing in this, community. This is a emotionally taxing space, and you handle it with humor and grace, which is remarkable. Really, an honor to be with you today. Thank you very, very much.
Yeah, it's been a pleasure having you. as always, we'll be posting this online and keep everyone up to date. We update the website and Instagram when there's new news from companies, if there's any press releases. So we'll be sure to share any updates that we get from Skyhawk. Thank you to the lovely Ashley as always, and really big thanks to you, Bill. And yeah, we'll see you soon.
Thank you so much.
There's lots of good news. Ashley, Lauren, take care of yourselves. Thank you very, very much.